My name's Shane Liddelow, and I'm the astrocyte guy. Astrocytes make up anywhere from 30 to 40% of all of the cells in your brain. When astrocytes are healthy, they provide nutrients or food to neurons, and they also take away their waste products. The discovery was that astrocytes, when they become reactive, can very actively and very specifically drive the death of neurons. It turns out that in every neurodegenerative disease that we have looked at, things like Alzheimer's disease to glaucoma, we see these dysfunctional astrocytes present. There's not a single therapy that targets dysfunctional astrocytes. One of the things the lab is able to do now is take what we learn in a laboratory setting and start applying that to different neurological conditions. Not only does it give us this sort of basic biological understanding of what's happening in the brain, the sole reason I got into neuroscience in the first place, but it also enables us to be first in class to actually produce novel therapeutics and help a whole wide range of people. I grew up in a small town, around 4,000 people, called Manjimup in the southwest of Western Australia. And it's a small farming town, lots of mining, forestry, things like this. When I started college at the University of Melbourne in Australia, in my first year I was mugged and I had an aneurysm. And in trying to learn what would be the outcomes of an aneurysm, I realized there was very little known about how to treat patients with neurological conditions. And so I embarked on a whole new avenue of study looking at neuroscience and biomedical aspects of science. One of the things that astrocytes are really good at doing is helping neurons to form networks across the brain. We were really interested in whether or not astrocytes would still do this when they were reactive or diseased. And it wasn't until hours of looking down a microscope I realized that not only were those connections not forming, but those neurons were dying in front of my eyes. Not only do these astrocytes make toxins that kill these neurons, but these astrocytes come in and remove those damaged neurons. This cleanup machinery is actually co-opted to be used to drive degeneration. Many scientists talk about the best thing about being a scientist is being, for a moment in time, the only person in the entirety of human existence that knows a fact. And so that was an incredibly exciting moment. The NYU Langone difference is the fact that not only do we have really excellent basic scientists working at the forefront of their respective fields, from neuroscience to immunology and genetics, but we also have the wealth of expertise on the clinical arm of the institution as well, where we can really readily understand how this is affecting patients. On any given day, you can bump into someone who has done neurosurgery that morning. We can immediately take that discovery or that insight from a quick chat over coffee into the lab and start digging into some new biology very quickly. So ultimately, it's really about our people, not just in the lab here, but also across the entire institution, that they're able to communicate across a common love of science and understanding these really complex problems. It's a really great place to do exactly these sorts of discoveries and to study the weird and wonderful cells of the brain. And I'm really excited about what the lab is going to discover next.